Hey everyone, welcome to unit number 7, non-visual senses. We're going to start off with talking about audition, our sense of hearing. So we're going to be breaking up our discussion of audition across a couple of mini lectures. First off, we're going to start by talking about the structure and function of our auditory system. Let's start off by defining the stimulus that we're dealing with audition. This is sound waves, right? Vibration of molecules that are in the air. So we have alternative condensation and rarefication, or pulling apart of these molecules, which produce a wave. So here we have sound waves, which are just compressions and rarefications of the air, right? Vibrations through molecules in the air. Right, so we can represent these vibrations as waveforms, and the physical dimensions of those waveforms will sort of be coded as perceptual dimensions that we can talk about. So the first off is pitch, which is to do with frequency. So frequency is basically how far apart the peaks are on this waveform. So a low frequency sound right here, with these peaks being far apart, will have a low pitch, right? It'll have a low sound. Whereas a high frequency sound with the peaks close together will have a high frequency sound, will be high pitched. So the physical dimension is the frequency, whereas our psychological quality that we assign to that is pitch. Likewise, we have loudness, which is the intensity or the amplitude of the waveform. So the larger the waveform is, the bigger these peaks are, the louder it appears to be, whereas smaller peaks are a softer sound. So the physical dimension of amplitude or height of the wave is coded as loudness. Finally, we have timbre, which is a measure of the complexity. We'll talk in a little bit more detail later on when we're talking about auditory perception about timbre, but basically it has to do with how complex the waveform is, right? When you're hearing something, it's typically, with the exception of a pure tone, it's not just a single waveform. There's a lot of what we call overtones, which are just sort of other frequencies that exist at the same time. Much like when you perceive light, it's very rarely a pure light stimulus of just a pure wavelength of blue, for example. When you hear something, it's very rarely just a simple stimulus. And we'll talk about that again in more detail later on. Okay, so let's talk a bit about the anatomy of the ear so we can get a sense for how this transduction process works. So first off, we have the tympanic membrane, also known as the eardrum. Displacement of this, so when this vibrates from those sound waves that we talked about, it will initiate our perception of sound. So this is the first step in our sound perception, right? Those vibrations being funneled in through our ear canal and vibrating our tympanic membrane. So that vibration is transferred through the tympanic membrane into our ossicles, which are these three bones in the inner ear, the malleus, incus, and stapes. So those vibrations are transferred through this solid matter, which in turn vibrate the oval window. So the oval window is an opening in the bone that's surrounding the cochlea, which contains a membrane which is connected to the stapes. So basically we have the transferring of vibrations from the air through the tympanic membrane, through the ossicles, now via the oval window into the cochlea. The cochlea is this awesome little snail-shaped structure, which is in our inner ear, which contains three fluid-filled chambers. And don't worry, we're going to zoom in and talk about this quite a bit more. There's a scala vestibuli, a scala media, and a scala tympani. Okay, so let's get into talking about our cochlea anatomy. So here we have a cross-section, which shows us the scala vestibuli, the scala media, and the scala tympani. And in the center, we have the organ of corti, which we're also going to be talking quite a bit more about. So the scala vestibuli is what connects to the oval window. So this is the sort of beginning of the transfer of motion into our cochlea. You can remember this because vestibuli has the same root as vestibule, right? Like an entry chamber that you would come into when you're entering a house or something like that. So vestibuli is sort of like the vestibule of the cochlea, right? It's the entry chamber. This is where the vibration enters the cochlea and begins to vibrate the fluid inside of the scala vestibuli. The scala tympani, on the other hand, is on the underside of this cross-section. So the vibration transferred into the fluid that fills this region will sort of transfer around, make its way around the apex of the cochlea, and make its way back around to the scala tympani. This connects to the round window. The round window is just sort of a membranous portion that allows for the displacement of fluid. So it's a place in the membrane that has some give to it. So that way this fluid can vibrate and allow the propagation of that signal. The last section we're going to talk about is the scala media, so it's sort of in the middle. This contains the organ of corti. The organ of corti is our auditory receptor organ. Don't worry, we're gonna talk much more about the organ of corti. All right, let's talk organ of corti. So the organ of corti is comprised of a number of different components that we're going to talk about. First, there's the basilar membrane. This is a flexible membrane forming the base of the organ, right? Hence, basilar membrane. So it's this flexible membrane here on which the hair cells rest. So the hair cells are auditory receptor cells, which contain cilia, or these little hairs that come off, right? So hence the name hair cells. Up top here, we have the tectoral membrane. This is a rigid membrane which will cover the hair cells, and it actually makes contact with the cilia. Okay, we've gone through enough now that we can talk about the actual auditory transduction process. So how do we take sound waves in our environment and convert them into neural impulses that our brain can understand? So first, we have our vibration of the eardrum, which results in ossicle movement, which will then move the oval window. So the vibrations are going to start moving the oval window, which is going to cause basically waves in this in the cochlea, right? There's going to be fluid displacement in the scala vestibuli. This will bend the basilar membrane. Fluid displacement will then extend around to the scala tympani. So fluid displacement will continue on propagating here, which will result in round window movement. So the whole point of transferring this motion into the fluid inside of the cochlea is to cause this wave to propagate in this fluid medium, which is going to cause the basilar membrane to bend. And the properties of this waveform are going to cause more pronounced bending at some regions than others, right? So here we have the sound wave coming in. It's going to cause a particular region of the basilar membrane to flex back and forth, which is going to exert mechanical pressure on the hair cells in this region, which is going to help us code that specific pitch. Okay, the last piece we need to understand when we're talking about auditory transduction is what's happening at the level of the hair cell and the cilia as it's being mechanically stimulated by the movement of the basilar membrane. So the cilia are located on an individual hair cell, right? So here's our hair cell and here's our cilia, these little things extending out on the top. These are connected via tip links. Tip links are an elastic filament that attaches the tip of one cilium to the surrounding cilium. So all the cilia here are connected by tip links, these little elastic filaments. So these little things are engineered in such a way to be very sensitive to the mechanical pressure of the vibration of that membrane, right? So when they're oriented in their resting direction, there's moderate stretching that allows some low-level activation. Movement away from that default state will result in a decrease in firing rate because the tip link will not be exerting any pressure on this channel. Whereas when it's stretched in the way towards the activation state, you can see here there's going to be more tension on this tip link, which will allow this channel to open. So when the force of the cilia moving causes this stretching pressure from this tip link to open this channel, we get influx of positive ions. And since we get potassium and calcium ions flooding into this cilia, that's going to cause depolarization of the hair cell. So that's it. That is the moment where we turn sound waves into an electrochemical signal that our brain understands. So this weird convoluted system of turning vibrations in the air into vibrations in bone, into vibrations in fluid, which are going to be waving around this membrane, putting pressure on these hair cells in very specific points of our cochlea. And when that happens, that mechanical pressure causes mechanoreception, right? A physical change opens or closes ion channels. So when this is pulled over, pushed away from its resting state towards its active state, the tip link will pull open this mechanical channel and allow ions to flow in, causing a depolarization of it. So that's it. That's it from start to finish. Okay, that's it for our structure and function bit. I will post a video in addition to this on Perusal, sort of a demonstration, a nice animated way of talking through the transduction process. So you can hear it a different way with much cooler animations. All right, that's it for our first unit. I'll see you next time.